Hi guys, welcome to the channel. Today's lecture is about phospholipids. Phospholipids are polar ionic compounds that are composed of an alcohol attached to an either diglycerol or DAG or sphingosine through a phosphodiester bond. Same as fatty acids, phospholipids are amphipathic in nature so that they have a hydrophilic head and a hydrophobic tail. The head consists of a phosphate group plus whatever alcohol attached to it, and a long hydrophobic tail containing fatty acid or a fatty acid derived hydrocarbon. So here is the head and here is a tail. Both are attached through a backbone. This backbone is an alcohol. We divide phospholipids into two classes according to the type of this backbone, which is either glycerol or sphingosine. If this backbone is glycerol, so glycerophospholipid is formed, or if the alcohol is sphingosine, sphingophospholipid is formed. Okay, how phospholipids are important to us? Actually, they are main structure components of our cell membranes. So in membranes, the hydrophobic portion is associated with non-polar portions of other membrane, such as glycolipids, proteins, glycerol, while the polar head or hydrophobic head extends outwards, interacting with the intracellular or extracellular aqueous environment with the surrounding water, so that the hydrophobic part, or here is a bilayer, so hydrophobic tail of both layers are facing each other, while the heads, which are the polar parts, face the outward or the surrounding, which is consisting mainly of water. As we said, there are two classes of phospholipids. According to the type of the backbone, we decide the type of phospholipid, so that a glycerol forms glycerophospholipid, sphingosine forms sphingophospholipid. Both play a role in generation of lipid signaling molecules. Glycerophospholipids constitute the major class of phospholipids. These are the phospholipids that contain glycerol as a backbone. All contain phosphatidic acid, or they are all derivatives of phosphatidic acid. What is phosphatidic acid? This is the simplest glycerophospholipid, and it acts as a precursor of other members of this group. Phosphatidic acid is formed of a tag attached to a phosphate group. This phosphate group is attached to carbon number 3 on the backbone, so that here are the 3 carbons of glycerol. Carbon number 1 is attached to a fatty acid. Carbon number 2 attached to another fatty acid, so a DAG is formed. Then at carbon number 3, phosphate group is attached, so that phosphatic acid is formed. through phosphatic acid, we can get other members of this group. For example, choline plus Pa gives me leucine, and so on. However, there are three members we are going to talk about in this lecture: cardiolipine, plasmalogens, platelet activating factor. Let's talk about cardiolipine first. Cardiolipine is formed of two molecules of phosphatic acid that are estified through their phosphate groups to an additional glycerol. So that cardiolipine is formed of three glycerols, two phosphate groups, and 4 fatty acids. Because there is a glycerol attached to 2 fatty acids and a phosphate group, so this is the first molecule of phosphatic acid. The other one is formed of 2 fatty acids attached to glycerol and to a phosphate group. These phosphate groups are going to be joined together through another backbone of glycerol. So here are 3 glycerols, 2 phosphate groups, and 4 fatty acids. Are they important to us? Of course, they are found in human cells and bacterial cells. In our cells, they are found exclusively inside the inner mitochondrial membranes, and they are important for the electron transport chain, which is important for respiration, so that we could generate ATP. Cardiolipine is found in bacterial membranes, so that they are antigenic, which means they can be recognized by antibodies, so that we could form antibodies against this bacteria, which is an advantage. Plasmalogens are phosphatic acid which is somehow edited, so that when fatty acid at compound number 1 of the glycophospholipid is replaced by unsaturated alkyl group attached by ether linkage to the core glycerol, this plasmalogen is formed. If you need to ask what this alkyl group is, actually it is a functional group of any organic compound or any organic chemical that contains only carbon and hydrogen atoms, which are arranged in a chain. The formula is CnH21 plus 1. Actually, this is an extra information. If you don't need it, then skip it. Okay. Do our body contain plasmalogens? Of course. There are derivatives of the plasmalogen. For example, phosphatidylcholine is abundant in the heart muscle, and phosphatidylethanolamine is abundant in the nerve tissue, so they are important to us. Platelet activating factor. here, both fatty acids at carbon number 1 and carbon number 2 are replaced. The fatty acid at carbon number 1 is replaced by a saturated alkyl group, while at carbon number 2 the fatty acid is replaced as or pi acetyl residue. Its importance sends impulse to surface receptors so that it triggers or induces thrombosis and acute inflammatory events, so that they activate inflammatory cells such as macrophages, neutrophils, and they help platelets to aggregate. In addition, they help the alveolar macrophages to generate superoxide radicals. Sphengomyelin, the second type of phospholipids. The only significant sphingophospholipid in humans is sphengomyelin. The sphengomyelin consists of amino alkyl sphingosine, which acts as a backbone, unlike glycerophospholipids where glycerol is the backbone. A longer chain fatty acid is attached to the amino group of the sphingosine through amide linkage to produce ceramide. This reaction occurs due to the condensation of palmitoyl-CoA with serine as CoA and carboxyl group of serine are lost. The product is reduced in a pH-requiring reaction into sphingosine, which is acylated at the amino group with longer chain fatty acid, as we said, and then desaturated to produce ceramide. Finally, the alkyl group at carbon number 1 is esterified to phosphorylcholine, producing sphingomyelin, as we said, the only significant sphingophospholipid in humans. Why it is so important? Since it's an important constituent of the myelin of nerve fibers, which is made by Schwann cells. The myelin sheath is a layered membranous structures that insulate and protect neural fibers, mainly axons of the central nervous system. Here is an important note: say that all cells except mature erythrocytes can synthesize phospholipids. However, triglycerides, which is another type of lipids, their synthesis occurs only in liver, adipose tissue, lactating mammary glands, and intestinal mucosal cells. Choline reutilization. Should we reuse choline if the phospholipids is degraded or nucleotides is degraded? Yes. Choline is considered as essential dietary nutrient with adequate intake. Choline is very important to be used in the synthesis of acetylcholine, which is a neurotransmitter, mainly for parasympathetic nervous system. Synthesis of choline needs 3 methyl groups from methionine. Remember, methionine is an essential amino acid which is deficient in diet, so reuse of choline prevents possible choline deficiency. Choline is a glycerophospholipid used in formation of glycerophospholipid, which is the PPC, or lecithin. In DPPC, both positions 1 and 2 on the glycerol backbone is occupied by palmitate. DPPC is made and secreted by type 2 pneumocytes. This is a type of cells found in the alveoli in our lungs, and it is a major lipid component of lung surfactant. This lung surfactant is very important to decrease the surface tension in the fluid layer, to decrease the pressure needed to re-inflammate alveoli. Remember that water surrounding the alveoli may cause this alveoli to collapse since this water has a high surface tension, so that if this surfactant was not there, due to high surface tension of water, the water molecules would combine together into a single point, causing the alveoli to collapse. So, existence of lung surfactant is very important. Respiratory distress syndrome in infants is associated with insufficient surfactant production or secretion, so that they are not enough. So that their lung is said to be immature, and their lung maturation may be accelerated by giving the mother glucocorticoids during pregnancy and shortly before delivery. However, if this didn't happen after delivery, we may administrate natural or synthetic surfactant to prevent and treat infant respiratory distress syndrome. Respiratory distress syndrome may occur in adults whose surfactant producing pneumocytes have been damaged, destroyed. This may be caused by an infection or trauma during a car accident, for example, that cause fluid to accumulate in the alveoli and so impeding the exchange of carbon dioxide gas and oxygen gas, leading to respiratory distress syndrome. Lung maturity of a fetus may be determined by the ratio of DPPC into comparison with sphengomyelin in the amniotic fluid. A ratio above 2 is an evidence of maturity.